Hi, my name is Maria del Mar Alvarez, and today I'm going to present you my seminary about blockade of CD93 in pleural mesothelial cells, fuels anti-lung tumor immune responses. First of all, a small introduction about what is CD93. So, it is a transmembrane glycoprotein and it plays a role in a cell-to-cell addition process. It is involved in the regulation of phagocytosis and apoptotic cells and the promotion of angiogenesis both in physiology and disease, and it is involved in host defense as an early B-cell marker and antibody. Next, we have the antitumoral response. First, the receptors of the immune system recognizes molecular patterns associated to dangerous things like cancer. It can react to specific antigens or against antigens associated to tumors. So, at last, it occurs the opsonization for the ingestion and destruction of the tumor cell. It is important to emphasize on the general objective. This study wants to prove that the blocking of CD93 in pleural mesothelial cells fuels and anti-lung tumor immune responses. In this study, they used surgical specimens of lung tumor tissues from MPES from lung cancer patients and blood from healthy volunteers, and lung cancer patients with anti-PD-1 therapy were obtained. All patients and healthy volunteers were informed of the use of their samples and signed consent from were obtained. So now I'm going to explain the methods. First, we have the Western blot. It is a laboratory technique used to detect a specific protein. On this study, they utilize it to identify tumor extracellular vesicles, TEVs. The total cells and EVS were washed with PBS, laced in SDS buffer proteins, and samples were separated, transferred into VVF membranes, and proved with the corresponding primary antibodies. Then we have the flow cytometry. It is a laser-based technique used to detect and analyze the chemical and physical characteristics of cell or particles. In this method, recombinant proteins and antibodies were generated to test which antibody was the most effective one to increase CCL21A in PPMCS. Then there is the PCR. These tests detect the DNA or RNA of a pathogen or abnormal cells in a sample. It was used with the same purpose of the flow cytometry. Last is the immunohistochemistry. It is a method that uses the antibodies to check of certain antigen markers in a sample of tissue. And this study was also used to generate recombinant proteins and antibodies to test which antibody was the most effective one to increase CCL21A in PPMCS. Next, we have the results. In the PCR, mice were intrapleural injected with LLC-EVS. After 24 hours, the pleural CCL21A messenger RNA levels were determined by real-time PCR. CCL21A is a protein involved in immunoregulatory and inflammatory processes. We can see that in the presence of LLC-EVS, the expression of the protein CCL21A is higher. These two are small chemical synthesized RNAs that mimic endogenous messenger RNAs. Then we have the Western blot. When we have the expression of PBS here, here, and here, there is also a more expression of CD93. But we see a variation on the other results. If the color is clearer, it can be because there's something blocking it. Next, we have flow cytometry. In the A, mice were intrapleural injected with LLC-EVS. After 24 hours, we can see the incorporation of EVS into the pleura. In V, mice were treated with LLC-Lucy cells and LLC-EVS. CTO-D was intrapleurally injected 2 hours before each EVA injection. The incorporation of EVS can be seen not in CTO-D but in DMSO. And at last, we have the immunochemistry. In C, CD93 and CCL21A proteins in PMSAS of LLC lung tumor bearing mice were detected. The CD93 and CCL21A has a pretty high expression. In F, mice were intrapleurally injected with LLC-EVS. We can see that in the presence of PBS, it's higher the expression of the mesothelin and CD93 than in the
LLC-EVS.
Then, in the discussion, we took 3 authors. They all have conclusions that agree with the article, and I also agree with them. First, Shani I et al. TEF comprise various components responsible for tumorigenesis or suppression. Then we have Chin Ji et al. V16F10 EVS reportedly inhibited anti-tumor immunity by activating PD-1 signaling in T cells via PDL1. Then, Sun ZQ et al. TAF-derived miRNAs have potential roles as biomarkers in tumor diagnosis. And at last, we have the conclusions. The first one is that the CD93 is known to be involved in various diseases, not just in lung tumor, but with a similar role. So, for sure, it is pretty important to keep researching about this protein to help with the types of cancer successfully, like this study. The second one is that TEFs are crucial for this type of studies. It's fundamental to keep investigating them for further research on different types of tumors so that we can help millions of people as health professionals. And then the last one is that these type of studies where they use meticulously the molecular biology are crucial so that we can help our patients. With this study, we learned that the investigations of these small particles like CD93 is a good path to find good outcomes for all the people with this type of diseases. Thanks!